cycle. So if you'd like to follow along, one of the slides has a figure from Voet, and Pratt, Fundamentals of Biochemistry, figure 14.6, and that has the entire citric acid cycle on it. So if you'd like to print that out and follow along, that would be a good study aid. So here we are in our where are we in my outline of catabolic pathways. We are here. This is the citric acid cycle. And you notice that there are lots of different ways that we can feed into the citric acid cycle. Breakdown of carbohydrates, lipids, and proteins will break down to form compounds that can enter the citric acid cycle. So this is a figure that I showed you a while back where we can see that we have the biopolymers that make up our food are broken down into 2 and 3 carbon intermediates. So the 2 carbon intermediate would be acetyl-CoA and the 3 carbon intermediate would be pyruvate. So we'll see in this lecture how pyruvate can be converted to acetyl-CoA. The acetyl-CoA then enters the citric acid cycle and the carbons in acetyl-CoA are oxidized to form CO2. So that's an oxidation, so you have to have a reduction. So the coenzymes NAD+ and Q will accept those protons and electrons to become QH2 and NADH, and those will then enter the electron transport chain and produce ATP by oxidative phosphorylation. That is the subject of the next chapter. So I just wanted to say at the outset, I'll use the term citric acid cycle. There are some synonyms, Krebs cycle after Hans Krebs, who elucidated it, or sometimes TCA for tricarboxylic acid because citrate is a tricarboxylic acid. So remember I showed you this figure a while back when we talked about glycolysis, and glycolysis, of course, is glucose to pyruvate, and in that pathway, the stoichiometry is not shown, but we form for every 1 mole of glucose, we generate 2 moles of NADH, and we talked about the importance of not stopping at pyruvate. The pyruvate is converted to lactate, or in some organisms like yeast, to ethanol. But in both of these steps, the NADH generated is regenerated to NAD+, because the NAD+ will need to serve as a reactant for the GAPDH enzyme in glycolysis. So the same thing's happening over here. In the citric acid cycle, the citric acid cycle will generate the NADH, and then the NAD+ will be regenerated, but not until electron transport. But it is important to regenerate that NAD+ so that we can use it in glycolysis. So comparing this pathway, this is aerobic respiration. We do need oxygen. We'll yield between 30 and 32 ATPs. Oxygen is required. It's not required for the other 2 pathways. It's a lot slower, but you get a whole lot more ATP. So in all of these cases, whether you transform pyruvate to carbon dioxide and water, which is a complete oxidation, or lactate and ethanol and CO2, in all of those cases, it is important to regenerate NAD+ for glycolysis to continue. So here's Hans Krebs. So he is the one who elucidated the citric acid cycle. So this is the final common pathway for the complete oxidation of fuel molecules. So by complete, we mean all of the carbons are completely oxidized to carbon dioxide. So carbon dioxide, if you remember the structure from general chemistry, we have 4 bonds to oxygen. That's as oxidized as a carbon can get. So generating carbon dioxide involves the complete oxidation of the carbon atoms in our fuel molecules. So it's a multi-step catalyst, so it returns to its starting position. The other thing we're going to find out later is that it also provides intermediates for biosynthetic reactions. Glycolysis does that too. We have to make sure we don't siphon off too many of those intermediates because we want to keep the citric acid cycle going. So I have a link to an animated figure that I'm going to show you now. So that's on our slideshow. So we learned in the carbohydrate chapter that glycogen can be converted to glucose, and in glycolysis, glucose is converted to pyruvate. And the next step is pyruvate has 3 carbons. One of those carbons will be completely oxidized to CO2, and then the other 2 carbons form acetyl-CoA. And we can get acetyl-CoA from glycogen and glucose as shown but amino acids also can generate acetyl-CoA, and fatty acids generate acetyl-CoA when they are degraded as well. So we have the way to funnel all of the biological macromolecules that make up our diet to form acetyl-CoA. And then acetyl-CoA enters the citric acid cycle, and the 2 carbons in acetyl-CoA are completely oxidized to CO2. All right, let's go back to the slideshow. So here is the complete citric acid cycle pathway. So it shows all of the reactants and products, it shows all of the structures, all of the coenzymes, all of the enzymes. It shows which reactions are reversible and which ones aren't, so we can figure out where the pathway is regulated. I'm going to just start the animated figure so that you can see it. I won't play the whole thing, but I'll just link to it so you can see it, and you can see where the URL is so that you can play it if you like. It is not opening for some reason. I'm sorry that my program doesn't seem to let me open up those videos, but I have links at the end, so I'll try again and see if I can do that. In any case, the links to all of them are posted on Sakai, so you should be able to access those. So going on, the citric acid cycle occurs in the mitochondrial matrix, whereas glycolysis occurs in the cytosol. So I thought we should have a review of mitochondrial structure. So the mitochondrion consists of an outer membrane and an inner membrane. The inner membrane is very, very highly folded. The inner membrane encloses a space called the matrix, and the space between the outer membrane and the inner membrane is called the inner membrane space. So the vast majority of the reactions in the citric acid cycle occur in the mitochondrial matrix. So that's that space enclosed by the inner mitochondrial membrane. So before we get started on all the details of the pathway, I wanted to point out that there are different forms of metabolic pathways. So we can have a linear pathway. Glycolysis is a linear pathway where you have a starting and an ending point. Some pathways are circular, and the citric acid cycle is an example of that. Some of them are repetitive, where like fatty acid degradation, where you remove 2 carbons at a time from a fatty acid molecule, and then you go through the pathway in several different iterations. So we'll talk about that in chapter 17. So today, we'll focus on circular pathways. Citric acid cycle is circular, and we will begin and end with OAA, or oxaloacetate. So oxaloacetate is used as a reactant in the first step, and it will be regenerated in the last step. So we have a bit of a problem here. So when glucose is converted to pyruvate, that happens in the cytosol. But we need to get the pyruvate into the mitochondrial matrix in order for the pyruvate to be able to produce acetyl-CoA to enter into the citric acid cycle. So that's not a problem for organisms that produce pyruvate from ethanol, or for our muscle cells that convert pyruvate to lactate. That occurs in the cytosol. So getting through the outer mitochondrial membrane is actually fairly easy. There are pores that will allow the passage of the pyruvate into the inner membrane space, but then the pyruvate needs to cross the inner mitochondrial membrane, and that is a lot more impermeable. So transporters are required in order for that to happen. So this is sort of a convoluted way of getting the pyruvate in, but this is how it works. So pyruvate is brought in through a pyruvate transporter, and then pyruvate will be converted to acetyl-CoA, and I'll show you that in a minute. Pyruvate in the mitochondrial matrix is converted to OAA. That's the pyruvate carboxylase reaction, so hopefully you remember that from gluconeogenesis. And as part of this coordinated transport system, the OAA is converted to phosphoenol pyruvate. That's the Pepsi-K reaction, and then the phosphoenol pyruvate leaves, and then phosphoenol pyruvate goes back into the cytosol. So that's the transport system that exists to carry pyruvate that's generated in glycolysis into the mitochondrial matrix. That's called the pyruvate translocase transporter. So this is the way to link these two pathways. So again, I have just diagrammed over here. Glucose forms pyruvate in glycolysis. Pyruvate's then converted to acetyl-CoA, and that's the link between glycolysis and the citric acid cycle. So pyruvate has to be converted to acetyl-CoA, and then we can begin the citric acid cycle. So the conversion of pyruvate to acetyl-CoA is a very important step in linking these two pathways. So let's remind ourselves of what coenzyme A looks like. So coenzyme A, the A stands for adenosine. So we have the adenine and the ribose and the phosphate right here. So this forms adenosine and 2 phosphates. This is pantothenic acid. It's a B vitamin that's required in your diet. And then this portion right here, this is a sulfhydryl group. So sometimes we say CoA-SH because the important chemistry happens at the sulfhydryl group. And what we do is we attach an acetyl group to this. So this is acetyl-CoA, and we form a thioester bond. And we should note that the hydrolysis of a thioester bond is an exergonic process that yields a great amount of free energy, about the same amount of energy as the hydrolysis of a phosphoanhydride bond in ATP. So that will be important in the energetics of this pathway. So let's look at this reaction. It's kind of complicated. The enzyme is pyruvate dehydrogenase, so we know from the name that it's a redox reaction because all redox reactions are catalyzed by dehydrogenase enzymes. So there's a lot going on in this reaction. We convert pyruvate to acetyl-CoA. We will lose this carboxylate group as a CO2, so that's the oxidation, and then we're left with an acetyl group that will be attached to coenzyme A via that thioester linkage. NAD plus in the process will be reduced to NADH. So NAD plus picks up the protons and the electrons lost when the carbon in the carboxylate group in pyruvate is oxidized to CO2. So the enzyme is pyruvate dehydrogenase. So this is a picture here of the enzyme. Even in bacteria, this is a very complicated enzyme. So it consists of a dodecahedron core of 60 E2 subunits surrounded by a protein shell. The eukaryotic pyruvate dehydrogenase is even larger, with somewhere between 42 and 48 copies of E1, 60 copies of E2, and 6 to 12 copies of E3 plus proteins to hold it all together. So in the next slide, I'll show you how each one of these enzymes, E1, E2, and E3, participate in the conversion of pyruvate to acetyl-CoA. So here is the diagram right here. So here's pyruvate, and it's shown here in red. And in the first step, the carboxylate group is lost as CO2. And the next thing that happens is this portion of the pyruvate molecule is now bound to TPP. So TPP is thiamine pyrophosphate. So thiamine is also a B vitamin. So a thiamine pyrophosphate is a coenzyme portion of the E1, so its proper name is pyruvate dehydrogenase, is E1. So the thiamine pyrophosphate carries this portion that's left of the pyruvate after we've lost the carboxylate group to CO2. And then this is transferred to an acyl lipoyl lysine group in E2. So this big green blob here is E2, so its name is dihydrolipoyl transacetylase. And this has also a coenzyme. This is an oxidized lipoyl lysine, so there's a what looks like a disulfide bond right here. So that is hydrolyzed when the acetyl-CoA is bound to that sulfur. So that's the second step. In the third step, we bring in our second substrate, the coenzyme A, and the coenzyme A then, this acetyl group then is transferred to coenzyme A to form the acetyl-CoA. So in the meantime, the next thing that happens is this lipoyl lysine group now is completely reduced. So it is now in possession of the protons and electrons that are lost in this oxidation of pyruvate to acetyl-CoA and CO2. So in the next steps, what we need to do is we need to regenerate that to the oxidized lipoyl lysine. And that happens in 2 steps. The protons and electrons are transferred to FAD, which is a coenzyme that's part of the dihydrolipoyl dehydrogenase in E3, and the FAD accepts those protons and electrons to become FADH2, and then finally those protons and electrons are transferred to NAD plus to form NADH. So you can see here what the reactants are. The reactants are pyruvate and coenzyme A, and the products are acetyl-CoA, carbon dioxide, and NADH and H+. So in all, 5 reactions, all catalyzed by this pyruvate dehydrogenase that consists of these 3 different types of enzymes. These are all required to convert pyruvate to acetyl-CoA. So we have acetyl-CoA now. So what happens next is the acetyl-CoA will condense with OAA, or oxaloacetate, in the first reaction to form citrate. So this is an overview of the citric acid cycle, and it shows that this is a redox pathway. So the acetyl-CoA carbons right here, those 2 carbons will be released as carbon dioxide, and in the process, protons and electrons are lost. And we will generate NADH, we'll regenerate 3 of them. So we have 1, 2, 3 molecules of NADH that are generated per turn, and 1 molecule of FADH2 that's generated. This is also an exergonic pathway, so we will capture some of that energy that's released in the form of GTP or in the form of ATP in some organisms, although we should note that GTP is energetically equivalent to ATP. So this is a redox pathway. So if we go back to where we started with glycolysis, remember we are on a journey from glycolysis to complete oxidation to CO2 and water. So that means that eventually all 6 of the carbons in glucose will be oxidized to 6CO2. And for complete oxidation, we're going to need oxygen. We'll need 6 oxygens, so 6 of those oxygens, or 12 atoms of oxygen, will accept protons and electrons to form 12 waters. So in this process, glucose is oxidized to CO2, and we're seeing that now. We've just lost our first CO2 in the conversion of pyruvate to acetyl-CoA, and then we'll lose the second and third CO2s with 1 turn of the citric acid cycle. We will need to go through 2 turns per mole of glucose because 1 glucose produces 2 pyruvates, and 2 pyruvates produces 2 acetyl-CoA. So in the process, we will lose protons and electrons. They will eventually be donated to oxygen to form water, but for right now, the coenzymes are in possession of those protons and electrons. So if we look at the fates of carbon, stoichiometrically, it is true that 2 acetyl-CoA carbons in will give us 2 CO2 carbons out as CO2. But if we label those, we find that it's not the exact carbons. So here in this picture, if we have acetyl-CoA as red carbons, if we followed the first steps and show that we lose 2 carbons here, notice that the carbons that we are losing are gray carbons. So it's not exactly the same carbons out as in, but stoichiometrically we have 2 carbons in as acetyl-CoA and 2 carbons out as CO2. So here is yet another picture that emphasizes that as the acetyl-CoA is oxidized to CO2, we are going to be gathering protons and electrons, and all of these protons and electrons are transferred to these carriers, NAD plus to form NADH and FAD to form FADH2. So this is a redox pathway. The carbons in acetyl-CoA are oxidized to CO2, and the NAD plus and eventually coenzyme Q will accept those protons and electrons and become reduced to NADH and H plus and QH2 respectively. So the energetics of this pathway. So far I've focused on how many carbons in, how many carbons out. where's the oxidation, where's the reduction? Well, this is a downhill or an energy producing pathway with an overall delta G of a negative number. So we're going to capture some of that energy in the form of GTP. But we should also note that NADH and QH2 can also be considered to be cellular energy currency because we will use those coenzyme molecules to generate ATP in the next chapter when we talk about electron transport and oxidative phosphorylation. Okay, so we have a great overview now, so we are finally ready to talk about each of those reactions in detail. So in the first reaction, oxaloacetate condenses with acetyl-CoA to form citrate. And here the artist has shown the acetyl group in red so that we can follow along. So here's citrate, and the carbons here in black come from the OAA, and the carbons here in red come from the acetyl-CoA. So we should note that the artists have drawn an arrow in one direction. So that means this is a reaction that only goes in one direction. This is an irreversible reaction. So the pathway will be regulated at this point. The reaction occurs only in this direction because we are going to, in the course of the reaction, hydrolyze an unfortunately named high-energy thioester bond. We probably shouldn't call it high energy because it's the hydrolysis of this thioester bond that is an exergonic process. So that's what drives this reaction forward, because normally taking two small molecules and forming one big molecule would not be energetically favorable. But with the hydrolysis of that thioester bond, we can make that happen. So the enzyme is citrate synthase. So just to keep our enzyme names straight, we are synthesizing citrate. So that's where the synthase comes from. There are other enzymes called synthetases, but those enzymes involve ATP, and this one doesn't. So this is called a synthase. So I wanted to look at a structure of the enzyme. So this is a citrate synthase, and there are two subunits. So here in this picture, there's one that's green and one that's tan, and each subunit undergoes a very large conformational change on binding OAA. So OAA binds first, and then that creates a binding site for acetyl-CoA. So we want to make sure that we capture that energy of the hydrolysis of the thioester bond in order to drive this reaction forward. So in this picture, the open form of the enzyme is shown by itself, and then the closed form is shown with bound OAA and a stable analog of acetyl-CoA. It's not acetyl-CoA itself, because then the reaction would take place, and if you're doing X-ray crystallography, you want the enzymes to sit still for you so you can take their picture. So a stable analog won't be hydrolyzed, so that allows us to take a nice picture of the citrate synthase with both of its substrates bound. So here's the next step. So citrate is converted to isocitrate, and that involves a dehydration and then a rehydration, and the intermediate is aconitate. So the enzyme here is called aconitase, and it is named for its intermediate, which is why I wanted to show it to you. So the reason we carry out this isomerization is that you might remember from organic chemistry that if you have a tertiary alcohol, that's not oxidizable. But a secondary alcohol is. So we need to convert the non-oxidizable tertiary alcohol to the oxidizable secondary alcohol that we see in isocitrate. We will not regulate the pathway at this step. You can see that the artist has given us that information by drawing us the equilibrium arrows in both directions. So this shows the next step. This is isocitrate being converted to alpha-ketoglutarate, and a little bit of the mechanism is shown here. So a lot is going on in this reaction too. So here we lose our first CO2. This carboxylate group on isocitrate is lost to form CO2, and that's an oxidation, so we have a reduction. So NAD plus will be reduced to NADH and H plus, and we will form alpha-ketoglutarate. So this is our first oxidative decarboxylation. So we oxidatively decarboxylate the isocitrate to form alpha-ketoglutarate. So we go from 6 carbons now down to 5. We've lost one of our carbons as CO2. This is a redox reaction, and the enzyme is isocitrate dehydrogenase. Remember that all redox reactions are catalyzed by dehydrogenase enzymes. So the alcohol group is oxidized. So if we look over here, this carbon right here, and the artist shows this in green right here, this carbon is bound to 1 hydrogen and 1 oxygen. Over here, you can see that the carbon is bound to 2 oxygens. Sorry, there are 2 bonds to oxygens, I'm sorry. So what that means is that the number of bonds to oxygen in the carbon have increased and the number of bonds to hydrogen have decreased. So that indicates an oxidation. So this is a redox reaction. So we've oxidatively decarboxylated our isocitrate. We have CO2. We have a carbonyl group where we had an alcohol group, and we have generated a CO2, and we have generated our first NADH coenzyme that has the electrons and protons lost during this redox reaction. So the next step involves another oxidative decarboxylation, and I should have pointed out that the last reaction was irreversible. This one is too, and what we're going to do is we're going to lose this carboxylate group as CO2, and I also highlighted this bond here because we're also going to form a thioester bond. So here we have another oxidative decarboxylation. We're going to lose this carboxylate group as CO2, and these 4 carbons now then become bound to coenzyme A. So we have succinyl-CoA. So we've lost our second molecule of CO2, and we have an alpha-ketoglutarate dehydrogenase complex. And we won't go through the enzyme mechanism, but you already know it because it's almost the same as the pyruvate dehydrogenase complex, because the same kind of thing is happening. You're losing a carbon dioxide, carboxylic acid group is carbon dioxide. You're forming a thioester bond between the carbons that are left and coenzyme A, and you're forming NADH. So the alpha-ketoglutarate dehydrogenase complex also consists of E1, E2, and E3, and will accomplish this transformation. This is an irreversible reaction, and this is a place where we will regulate the pathway. So had we been in class, I would have asked you a top hat question, because I would have wanted you to see that this is a reaction that we call substrate level phosphorylation. So remember, we said there are 2 terms that we use to describe the synthesis of ATP and its equivalents, and that is oxidative phosphorylation, which we'll talk about in chapter 15, and substrate level phosphorylation. So in this reaction, we will capture some of the energy in the hydrolysis of this thioester bond right here and use it to couple GDP and phosphate to form GTP. So this involves GTP, which is energetically equivalent to ATP. So again, notice that this is a synthetase, so we involve ATP or its equivalent. And here we synthesize GTP. So the citric acid cycle as a whole is an exergonic pathway. So this is a way that we can capture some of its energy in GTP. So I wanted to show you a little bit of the mechanism of this enzyme because I thought it was interesting. The phosphate right here, which is one of the substrates, what happens is that you get nucleophilic attack right here, and then we release the coenzyme A. And now we have succinyl phosphate. So our succinyl-CoA, it's not succinyl-CoA anymore. The succinyl group is now covalently bound to phosphate and not coenzyme A. So we've swapped the coenzyme A for a phosphate. And notice we have this mixed anhydride right here. So the next thing that happens is nucleophilic attack of a histidine residue on the enzyme. So this is unprotonated and it serves as a nucleophile. Nucleophilically attacks this phosphate. And now the phosphate is covalently bound to the histidine. So we release succinate, and now that phosphate is transferred from the histidine to GDP to form GTP, and we've regenerated our enzyme, which has a histidine in its active site that's not protonated. So here you can see the sequential release of each one of the products. Okay, next up, succinate is converted to fumarate. So you can see that this is also not a this is a reversible reaction. We are not going to regulate the pathway here. This reaction is interesting. It's a redox reaction, we can tell, because the enzyme is succinate dehydrogenase. And also, if we look at these carbons right here, these carbons are bound to hydrogens, but if we look over here, we form a double bond. So we've decreased the number of bonds to hydrogen. These hydrogens and electrons are converted to, they are transferred to FAD to form FADH2. The way that this works is that the succinate dehydrogenase enzyme has a covalently bound prosthetic group called FAD, and that's showed maybe in a little bit more detail here. So here's the succinate dehydrogenase enzyme. So here we just say E for enzyme. And it also has a histidine, and but here there's a covalent bond between the nitrogen on the histidine of this enzyme to FAD. So that means in this reaction, when succinate is converted to fumarate and the enzyme catalyzes that reaction, the two protons and electrons on succinate are transferred to FAD to form FADH2. So those protons and electrons will be transferred to this ring form right here. So succinate is oxidized to fumarate. The protons and electrons are transferred to FAD to form FADH2. The next step that will happen is the protons and electrons will be transferred from FAD to coenzyme Q to form QH2. So that's why some of the diagrams of the citric acid cycle that you see, sometimes they'll say FAD and sometimes they'll say QH2. But FAD is formed, part of the prosthetic group of the succinate dehydrogenase. It accepts the electrons and protons first and then transfer them to Q to form QH2. So this enzyme, the succinate dehydrogenase, is located in the inner mitochondrial membrane. So it's the only one that's not in the mitochondrial matrix. All of the rest of the citric acid cycle enzymes are in the mitochondrial matrix. This is going to become important in the next chapter when we talk about electron transport. So just file that away in your brain for now. I wanted to show you the oxidized and reduced forms of FAD. So here's FAD. If we look at what's highlighted here in blue, when protons and electrons are transferred to FAD, what happens is the electrons and protons form part of this ring structure right here. So we form bonds between the nitrogens and the two hydrogens. So the hydrogens are shown here in pink. And we form new bonds between the nitrogens and the hydrogens. So that's the reduced form of the FADH2. This reaction doesn't show this, but the FAD is actually capable of accepting one proton and one electron at a time. Not everybody can do that. NAD plus, when it forms NADH, can't do that. It has to accept both electrons at a time. But FAD can accept one electron at a time. So that's going to be important later on too. Okay, next up, fumarate is converted to malate. The enzyme is fumarase, and this shows the carbanion transition state. So what we're going to do is we're going to hydrate fumarate to malate. And when we do that, we essentially add water trans across the double bond, so we only form the L isomer. And this reaction has a negative delta G, as you can see here in the figure. This is a reversible reaction, so this is not a place where we will control the pathway. All right, we finally gotten to the end. Malate is converted to oxaloacetate and the malate dehydrogenase reaction. If you look at this carbon right here, this carbon has one hydrogen, one bond to hydrogen, one bond to oxygen. And if you look over here, we have two bonds to oxygen, so the malate is oxidized to oxaloacetate. This involves the transfer of two protons and two electrons to NAD plus to form NADH and H plus, and it is a redox reaction because we have a dehydrogenase enzyme. So we have an oxidation of malate to OAA. The reaction is reversible. We won't generate the pathway here, and we've regenerated OAA. So this completes the cycle, and we can add another molecule of acetyl-CoA and start the whole thing all over again. So this is the overview of the citric acid cycle. We started out with acetyl-CoA, and the two carbons in acetyl-CoA were oxidized to CO2, so we would simply exhale that. We captured some of the free energy in the conversion of acetyl-CoA to CO2. When we captured some of that energy as GTP, which can be used to do cellular work. The oxidation of acetyl-CoA to CO2 involved the loss of protons and electrons, which were picked up by NAD plus and coenzyme Q to form NADH and QH2 respectively, and those will be oxidized in electron transport. So here's our overview of the citric acid cycle. We can see acetyl-CoA comes in, two CO2s come out. The CO2s were oxidized, so we form per turn of the cycle 3 NADHs, 1 FADH2 that will donate its electrons to Q to form QH2, and we've formed 1 GTP in substrate level phosphorylation. So I clicked on a link. I need a flash player for this. This is a guided exploration that will allow you, if you want to go back and look at any of the reactions individually, you can click an arrow right here. So if I'm going to click the first arrow down here, it shows a little bit of the mechanism.
The condensation of acetyl-CoA and oxaloacetate to form citral-CoA.
I'm going to go back to, I actually closed that animation because I found out that trying to record the animation with the voiceover gives you something that's very echoey. So anyway, you can see the URL in that, when I first turned it on, there is a link to that animation on Sakai, so you can see it there. But what it allows you to do is to look at each one of the reactions, and it'll play out a little bit of the mechanism for you. Here's another animation. This animation, oh, that's the one I showed before. So let's go back to, okay, so in this animation right here, this shows the entire citric acid pathway. And again, it will sort of step everything out for you, and you can look at each one of the reactions. So since I've already gone over that, I won't go over the entire animation here. But what's nice about this is that it labels the carbons for you. So if you see there's a little double dagger right here, you can follow the carbon as it goes around the cycle. So again, if I just show the first part of it, it shows acetyl-CoA entering the citric acid cycle, condensing with OAA to form citrate. And there's another label here. So here's a little star label right here. So when we have the citrate, we have the double dagger. So that's one of the carbons that came from acetyl-CoA. And then this carbon came from citrate. So if you do this animation and follow the whole thing, you'll be able to find out what happens to those carbons in one turn of the citric acid cycle. So we do always want to talk about regulation of the pathway, and that is going to be the subject of the next lecture. But I still wanted to point out to you that I wanted to remind you that you can form acetyl-CoA from pyruvate that was obtained through glucose, because we've been talking about carbohydrate metabolism. But we will find out in later chapters that amino acids can be broken down to pyruvate and acetyl-CoA, and lipid molecules are broken down to acetyl-CoA too. So in these reactions, acetyl-CoA is formed, so the entry into the cycle is largely controlled by the acetyl-CoA concentration. So for our next lecture, we will start with how do we control the pathway? So if we'd been in class, I would have shown you a top hat question, and hopefully you would have looked at the delta G's, not the delta G zero prime, but the delta G's, and we don't have the exact values provided for us, but the author does show us that we have the citrate synthase, the isocitrate dehydrogenase, and the alpha-ketoglutarate dehydrogenase reactions are all irreversible. They have negative values of delta G. So that's a good way for us to control the pathway. So whenever you're looking to control a pathway, remember we're always looking for those large negative delta G values. But we also have to control the entry into the cycle. So when we talk about the regulation of the citric acid cycle, we're also going to talk about pyruvate dehydrogenase because that controls how much acetyl-CoA we have. So even though the pyruvate to acetyl-CoA transformation isn't strictly part of the citric acid cycle, we have to talk about its regulation as part of regulating the pathway. So I'm going to leave you with another video. I'm just going to click on this very quickly, and I'm going to close it again because, again, it's going to form this echoey sound in my screencast program. So this is a link to a citric acid cycle video on YouTube, and it is made by our friends at Garland Science Publishing, and there it is.
Cells break down food molecules, such as glucose, through multi-step pathways.
So I closed it again because I found out that if I tried to do this, you get this very echoey sound in my screencast program. But there's a link to this on Sakai, and hopefully you were able to capture the URL if you want to visit it. But I wanted to tell you about this video because there's a very amusing part of it, that what happens is the narrator will go through all of the steps of the citric acid cycle and show a little bit of the mechanisms too. But in the background, a classical guitar player is playing this song called Oranges and Lemons. So this is not a song that is very popular in the United States, but I spent a year in London with my twins when they were 3, so I learned a lot of British nursery rhymes. So this is one of them. So it's called Oranges and Lemons Say the Bells of St. Clement's. So these are all the bells of the churches in London. But the pun here is that oranges and lemons, the tie to the citric acid cycle video, because of course oranges and lemons have citric acid in them. So it is too early in the morning for me to sing this for you, so I will not do that. But if you want to watch that video, and if you're wondering what the classical guitar player is playing, that's what's being played. So that's it for today. So go ahead and read this section in Essential Biochemistry and do the relevant problems. Here are my references, and I will see you next time.